guys, our topic is Gaucher's disease. So let's see the discovery first. Gaucher's disease is discovered by Philip Gaucher in the year 1882. So in this disease is actually accumulation of glucocerebrocyte. Glucocerebrocyte is there on the cell membranes. Actually when the cell dies, it has to be taken up by the macrophages. The macrophages have lysosomes. The lysosomes are supposed to destroy it. But due to the fault in the enzymes, they are not destroyed properly and they get accumulated in the macrophages.
So usually what happens is that glucocerebrocyte moves to the macrophages, and over there in the lysosome there's an enzyme known as glucocerebrosidase which is used to break down glucocerebrocyte into glucose and ceramide. This is the usual metabolism, but in the case of Gaucher's disease, there is a deficiency of glucocerebrosidase enzyme, and due to this, there is an accumulation of glucocerebrosidase in the lysosome. Now, this accumulation can be seen. The cells, due to this accumulation, have a crumpled tissue paper-like appearance, like this one right here. And this is a characteristic feature of Gaucher's disease and can be easily seen through a microscope. So now due to this accumulation, there is increased secretion of lysosomal enzymes as well as pro-inflammatory cytotoxins, which what they do is they cause inflammation and cause tissue scarring in many organs and organs such as the liver and spleen. And this has many metabolic effects, and it causes anemia, it causes osteoporosis, and also causes thrombocytopenia. Now moving on to the types.
So coming to the classification, we have three types of Gaucher's disease: type 1, type 2, and type 3. Talking about the type 1, it's the most commonest type of Gaucher's disease, and it's usually asymptomatic, but some people few symptoms are. And organs which are primarily affected by this type are liver, bone, and skin, causing hepatosplenomegaly, osteoporosis, anemia, and WBCs are also affected, causing leukemia. And for treatment, enzyme replacement therapy is effective for this type 1 Gaucher's disease. And since there is no neurological symptoms are present, it's also known as non-neuropathic Gaucher's disease, and that differentiates type 1 from other types of Gaucher's diseases. Now, what is type 2 Gaucher's disease? This is the rare and severe type of Gaucher's disease, which affects the brain and other organs of the infants and newborns. Usually, it has the same symptoms as type 1. And it also has symptoms of neurological symptoms, causing severe brain damage, seizures, developmental regressions. And for the treatment, there is no treatment for Gaucher's type 2 Gaucher's disease. But for the symptoms, to cure the symptoms, we can have in supportive therapies. And this is also known as acute neuropathic Gaucher's disease.
Now moving on to the type 3 Gaucher's disease, also known as chronic neuropathic Gaucher's disease. Its severity is between type 1 and type 2, and its symptoms develop at slower rate compared to type 1 and type 2. And it has both the symptoms, neurological as well as visceral symptoms, like hepatosplenomegaly, anemia, brain damage, neural tissue damage, and seizures. Now moving on to treatment and diagnosis.
Before going to the diagnosis, let's talk about the GBA1 gene, which encodes the lysosomal enzyme called glucocerebrosidase. If any mutation in the gene causes two diseases, one is the commonest one, Parkinson's disease, and the second one is Gaucher's disease. Let's ask the question how to differentiate these two diseases. The first one, Parkinson's disease, which will develop neurological symptoms like resting tremor or gain, etc. But in all Gaucher's disease, in type 1, there won't be any neurological symptoms. And in Parkinson's disease, one more feature called, there will be accumulation of glucocerebrosidase and one more lipid called glucosylsphingosine. These accumulation causes the brain to produce a protein called synuclein. So the presence of synuclein are the only feature present in the Parkinson's disease.
Now about the diagnosis of the disease, we can diagnose the disease by the different way. First by the glucose-riboside activity. There is a test is known as beta glucoside test. In this test, we can identify the enzyme activity is high or low. By this way, we can diagnose the disease, and then by the way of genetic testing. In the genetic testing, seeing the mutation in the GBA gene, we can diagnose the disease. And last by the clinical symptoms and clinical history and symptoms, and the presenting symptoms like bone pain, swelling, and enlargement of organs. Enlargement of organs like in liver, it causes the hepatomegaly, and splenomegaly, and in bone it causes the osteoporosis. And then by the treatment, and there is two treatment to cure the disease. First one is enzyme replacement therapy, and the second one is substrate reductive therapy.
So last we have made a mnemonic chart for this disease. So the main enzyme, that is the glucose cerebrosidase. For that, a man standing with the glued cerebrum on its side we have an AS card, which means the glued cerebrosidase, which represents the glucose cerebrosidase enzyme. So on the side of the chart we have a glued cerebrum on the side. So which denotes the increased cerebrocytes, which leads to the Gaucher's disease. So we have an image depicting the hepatomegaly and splenomegaly, and on the side we have placed a pan, which represents the pancytopenia, and last we have a skeleton system, the Gaucher, so which represents osteoporosis.